I was not prepared to introduce the DFW for today. Raul, can you please do? Oh, we have lost Raul, I think.
Have we? Okay, well, I can carry on with the presentation if someone's able to move it forward for me. So we should be on the first slide, which is the introduction to the Doctors Federation for the World, which is an international organization that has spanned over 5 years and has played a relentless and productive and important role in 5 continents. The DFW is built on hope and broken paradigms. So next slide, please. Are we able to move that forward?
Yeah. Yeah.
Thank you. So the DFW was founded in 2020 by Dr. Famny Casallo. The DFW is a non-profit organization which has been endorsed by numerous health specialists from all over the world. Our aim is to address acute and chronic diseases from a pathophysiological viewpoint with a long-term approach in which people from 5 continents participate on a voluntary basis. Our innovative, disruptive and constructive strength has enabled real-time interdisciplinary communication and work from health specialists with simultaneous translation into 8 different languages, which are English, Spanish, Italian, Portuguese, French. And today we also have Hindi and Arabic. Next slide, please. Our mission
Nuestra misión se centra. Oh, Vivi, si me puedes pasar a la cabina de español, de español, por favor. Gracias. Go ahead, Rachel.
Okay, we're on slide 3. Our mission is focused on providing integrative health solutions for human well-being. Our vision is to become the leading international organization in the search for safe, accessible, effective, and efficient treatments and preventative interventions. Our values seek open equality of ideas and humanity. Our objectives focus on the generation of solutions of long-term value, as well as close collaboration with clinicians, researchers, and the general public. Finally, our activities have focused on both the communication of monthly preventative health schemes, as well as interdisciplinary work with authorities and governments. Next slide, please. Our physicians around the globe have been using evidence-based medicine approaches to give feedback to their governments, societies regarding preventative and curative schemes. Next slide, please. We have held 119 lectures with the following profile of speakers: 59 with doctorate level education, 41 with master's degree or other speciality, 25 with bachelor's degree, and with respect to our worldwide influence, the DFW is held in 59 from America, 37 from Europe, 11 from Africa, 15 from Asia, and 5 from Oceania. Next slide, please. The DFW is as a proactive organisation that is recognised for its deep proactive approach to community and long-term collaborative work with governments. It is respectful of local health decisions in each country. It also seeks no confrontation or the deterioration of any status quo or the disruption of current health agendas. Next slide, please. We focus on doctors, academics, researchers, and other health professional allies. Next slide, please. In addition, we target government authorities, health systems, regulatory offices, social media, intellectuals, philanthropists, and civil society. Next slide, please. Thanks to the above, in 2022, we managed to integrate our process in a successful and unprecedented international summit that took place virtually from November the 11th to the 15th in 2022. Thank you to everyone for your participation. We integrated the most transcendental academic program in the history of mankind. It was the largest, with 127 speakers from 73 countries covering the 4 pillars. This was measurable It had our social actions. It was covered on Zoom, Facebook, YouTube, and LinkedIn, and it was multilingual in 5 languages in simultaneous translation. So a great achievement all around. Next slide, please. Our management is based on 4 main pillars, which are clinical, practical guidelines, traditional and integrative medicine, impact on communities at risk, and public policies in crisis situations. Next slide, please. Finally, the DFW is concentrating on strengthening its structure, consolidation, its objectives, supporting and receiving support from clinicians, researchers, and academics, sharing our vision, mission, and values, as well as joining forces with like-minded medical associations. We invite you to be part of our family and help us with your donation to support our technological structure and translators. Next slide. At DFW, we are convinced that by only dreaming big, working tirelessly and staying focused and working while surrounded by valuable people like yourselves, we will achieve and exceed our goals. Thank you, and thank you for joining us here today. Okay, so DFW is a non-profit organisation where everyone receives no payment at all. During 5 years of our structure, we have been supported from grants of its board of directors only. We kindly ask you for your support in making sure that your microphones are switched off for the presentations throughout the whole session in order that we do not exceed 2 hours. There will be no live questions for our keynote speaker, but you can interact with her throughout the chat box within these 8 different languages. To our speaker, we kindly remind you to keep on track with the maximum total 45 minutes and following a 30-minute round of questions and answers. We also remind you to speak as slowly as possible to allow our interpreters to translate properly. Now, let me introduce you to our amazing Vice President, Dr. Susan Raj from India, who will give us a short message, and she will be our Master of Ceremonies today, as well as introducing our guest of honor. Welcome, Dr. Raj.
Thank you very much, Rachel, and it's so nice to have Joachim here after a long time. Wonderful. And it's my privilege to be here and to give a keynote. Yes, we at the Doctors Federation for the World, we are working on integrative approach to all kinds of diseases. Yes, and we believe that our cells are designed to detox itself because we live in toxins. So our cells do perform certain tasks which has got entry and exit, and before exiting it has got its own processes. Yes, and those processes are studied by so many people in different ways, and like our ultimate purpose is to ensure that we have diseases coming in and getting out of our cells, not staying in there. So we believe that there is only one disease called toxin, and therefore there is only one remedy called detox. And this detox can happen in so many ways. And what we identify is that there are 7 methods by which our cells detox itself. So today, again, we have another eminent speaker with us, Dr. Stephanie Senev. And it is my privilege to introduce her and to fetch from her the entire knowledge about how to maintain this health, because health is the first thing we need to achieve in life so that the prosperity and our happiness sustains. That is why we have got so many specialists coming in and speaking to us. Today, we have got Dr. Stephanie Senev going to talk about deuterium, glyphosate, and glycocalyx. Dr. Stephanie Seneff is a senior research scientist at MIT's Computer Science and Artificial Intelligence Laboratory, CSAIL. With a background in electrical engineering, she has made significant contribution to the field of natural language processing, speech recognition, and human-computer interaction. She has authored numerous papers and is known for her interdisciplinary work, applying computational methods to study complex biological systems. Dr. Stephanie Seneff holds degrees from MIT, including a PhD in electrical engineering and computer science, and has a long-standing research career at one of the top technical institutes globally. It's such a privilege to have you here with us, Dr. Stephanie Seneff. The stage is yours. Thank you very much.
Thank you for that kind introduction, and we will just get started here. I was expecting an hour, so I may have trouble finishing in the time that we're expecting, but let's see how that goes. I will try to be brief. You can see my slide here. Okay, so I think I'm ready to go. Let me back up one. This is the title, Deuterium glyphosate and the glycocalyx. I'm very excited about this topic today. I have become I think the word is obsessed with deuterium lately because it's making so much sense to explain why glyphosate, which is Roundup, the active ingredient in Roundup, is so toxic to our organism. I like to start with a quote: "Science is a way of thinking much more than it is a body of knowledge," by Carl Sagan. Here's an outline of my talk. I'm first going to introduce deuterium, what it is and why it's important. I'm going to talk about sulfate, which is a topic that goes way back in my research, and the glycocalyx. Then I'll introduce glyphosate and explain why it is much more toxic than we believe. My theory about glyphosate's reasons for being so toxic is this substitution for glycine during protein synthesis. I'm going to get into some diseases, colitis and colon cancer, histamine intolerance, impaired fatty acid metabolism, and finally, a summary. So here's the introduction. Here's the big picture. Deuterium is a natural heavy isotope of hydrogen. It's present in the blood at about 5 times the concentration of calcium, so it's not trivial. It breaks the ATPase pumps in the mitochondria, which causes energy loss and the generation of reactive oxygen species. And mitochondrial dysfunction is linked to many, many diseases. There's a molecule called nicotinamide adenine dinucleotide, NAD, very important molecule in reactions, all kinds of biological reactions. It's a major carrier of a deuterium-depleted hydrogen atom when it becomes NADH. Gut microbes synthesize deuterium-depleted nutrients for the host. This is huge, and I think this is a big reason why we're sick. They're not making enough of these deuterium-depleted nutrients. Sulfation of the mucins, so sulfomucins in the gut, and in the glycocalyx, lining all the blood vessels. That is so important for the health of the circulation and the health of the gut. And it supports trapping of deuterium in gelled water. This is a theory that I have, and I have reasons to believe it from the standpoint of physics. Glyphosate suppresses many proteins that are involved in deuterium homeostasis. This is huge, and we'll get to more to that later. So deuterium heavy hydrogen. Hydrogen has 1 proton and 1 electron. And deuterium is the same, except it also has a neutron, and that makes it twice as heavy as hydrogen. So it's an isotope of hydrogen, just like carbon-14 is an isotope of carbon. It's everywhere in nature. It's present in ocean water at 155 parts per million. And it has distinct physical and chemical properties compared to hydrogen, which is what makes it very important, very interesting. Fats are a low deuterium nutrient, and I think that is probably a primary reason why fats are a healthy food. Deuterium management in the body involves trapping deuterium in gelled water outside the cell and invoking specialized enzymes that choose hydrogen over deuterium for their reaction in order to fuel the mitochondria with hydrogen rather than deuterium. The body obsesses on keeping deuterium out of the mitochondria. So there's some interesting work that was done in the 1960s where they had figured out how to make heavy water. And they thought, well, what happens if we give this heavy water to the mice? They had no idea what would happen. And apparently it tastes very much like regular water. So this is D2O instead of H2O. All the hydrogens have been turned into deuterium. And they got a 5% dextrose solution of this. Here's some quotes from that paper. Their appetites were voracious and constant savage attacks upon each other were continually in progress. Eventually, at varying intervals, the animals became hypoactive, lethargic, and dramatically weak. And death occurred in all cases within 6 to 10 days. So extremely poisonous. So here's the situation with the mitochondria. Very interesting. ATPase is a very interesting system that makes the ATP very essential in the mitochondria for producing the energy for the cell. The numbers are mind-boggling. 15,000 ATPase pumps in a single mitochondrion. There's a proton motive force that rotates the pumps, and that's what it gives you the energy to make the ATP. 1,000 cycles per second. Deuterons get stuck, and that's the main. It's like putting sugar in the gas tank, and that produces a stutter, and eventually that causes things to back up, causes reactive oxygen to spew out, and makes it the pump inefficient, and can eventually kill the pump. You have to make another one. So they also disrupt the proton-coupled electron transport, which is based on proton tunneling. Deuterons do not tunnel well, and so this causes decreased production of ATP and increased production of reactive oxygen, damaging the pump. So that's crucial. Here's a really interesting paper looking at deuterium in water. I was very surprised that they could find a very strong statistical significance to this result, where they looked at each state for all the states for which they had information, looking at the rates of depression and looking at the level of deuterium in the drinking water supply. So amazing to me that they got this very strong correlation saying that excess deuterium in the water leads to depression. Now, so I'm going to start to introduce sulfate here. Sulfate's critical role for maintaining exclusion zone water. And this is a lot of the work is Gerald Pollack's work. He's a good friend of mine. He has done fascinating work over many, many years on the glycocalyx and gelled water. So the glycocalyx is what lines all the blood vessels, and it releases protons and creates a battery. And so Gerald argues that that is a major energy source for the body. So my hypothesis is that the glycocalyx traps deuterium and extrudes deuterium-depleted protons. So the protons that it pushes out are depleted in deuterium, and that means it's pushing out into the blood supply, a low deuterium source of protons. Sulfate is crucial for maintaining the gel in the gelled water. And then there are also the sulfomucins that line the gut, and they serve a similar purpose to keep deuterium out of the water inside the gut that's involved with all the metabolism that's going on there. Sulfated glycosamine glycans become depleted in sulfate when you are chronically exposed to glyphosate. And I talk a lot about this in my book, which we'll get to, I think, here in a moment. So I have a preprint that I published, Is Deuterium Fractionation a Major Controlling Factor in Human Metabolism and Cancer? And in this preprint, I have this diagram to illustrate my thoughts on the glycocalyx. So here you have these endothelial cells lining the blood vessel. You have the gel here, which is all these glycosaminoglycans, gelled water, very slick border along the gel so that the red blood cells can just very smoothly slide through the capillary. And then the protons are being pushed out to create deuterium-depleted vascular water and deuterium-enriched gelled water. And that's a very important system for keeping deuterium out of the mitochondria. This is absolutely fascinating to me and extremely central to my hypothesis. The bugs in the gut make hydrogen gas, and they use that hydrogen, other bugs use that hydrogen gas to make organic matter. And in fact, for example, the short chain fatty acids. This system is central to providing the body, the organism as a whole, with deuterium depleted nutrients. So this hydrogen gas that's produced by these microbes is 80% depleted in deuterium. Deuterium stays with the liquid phase. This is very, very important. Part of it is going to be that the enzyme has special properties, but also just that deuterium always stays in the more dense state, from the solid to the gel to the liquid to the gas. Deuterium stays behind. So by the time you get to gas, we've got very low deuterium. So that some of the microbes are making this hydrogen and other microbes are using it to reduce carbon dioxide and to produce all kinds of interesting metabolites, including, for example, methionine, NADH, this is this deuterium-depleted H that I mentioned earlier, acetylcoenzyme A, which is what fuels the mitochondria. So all of these are really important nutrients that are coming out of that hydrogen gas by virtue of the activities of the gut microbes. So this was the first paper I read on deuterium. Laszlo introduced me to deuterium in December of 2019, and I jumped on it right away because it became very clear to me very quickly that glyphosate would disrupt this system, because I knew a lot about glyphosate at that time. Ketogenic diet and DDW health benefits. Deuterium depleted water is essential for mitochondria. People whose water supply is naturally depleted are healthier. Deuterium depletion maintains strong hydrogen bond networks to keep DNA stable. Deuterium depleted water inhibits tumor progression, and there's some great success stories around using DDW to treat cancer. And the ketogenic diet is a deuterium depleted diet, high fat diet. Glacier water is naturally depleted in deuterium because ice traps deuterium. So I think, for example, if you're hanging around a waterfall or near the ocean waves, the water that's leaving, the mist that is in the fog, these things are going to be low deuterium because of this principle that deuterium stays with liquid form. So I think this is why people have found that hanging out near a waterfall is therapeutic. It could be the reason is because you're breathing in deuterium depleted water. So sulfate in the glycocalyx, this is the glycocalyx here. Here's all the way around the artery. It's all about the blood. We are 2/3 water by mass and 99% by molecule count. Most of the water in the body is gelled. The big exception is the flowing blood. So the body has to keep the blood flowing, but needs to gel the water everywhere else. Blood delivers nutrients to and removes waste from all the tissues. Gelled water lines the blood vessel wall and provides slick, frictionless passage for the red blood cells, so you get low resistance, and therefore you can keep your blood pressure low. You don't have to push as hard. Sulfate keeps the water gelled along the border, and its transport is problematic, and this is especially true in the case of glyphosate. So sulfate is crucial to maintain structured water. Most cells maintain an extracellular matrix, which is similar to the glycocalyx, formed from proteins with complex sugar chains attached to them. Sulfate ions bind to the sugars at strategic locations in an irregular but not random pattern. The glycocalyx that lines the walls of all blood vessels is formed from these sugar protein complexes. And also the sulfomucins lining the interior of the gut barrier is another important example. So here's just kind of a schematic of the endothelial glycocalyx. Here's the glycocalyx. Here are the red blood cells coming through. This is an expanded look at this, what's in here, and you can see all these different activities going on. You have this is a glycosaminoglycan chain, GAG chain here attached to the proteins. And then you have various enzymes, EK-SOD and AT3 are various metabolic activities that are going on. Cellular signaling mechanism. So this is an antioxidant, and this one is a clot protective. These two guys are there hanging out in that on that glycocalyx. So when you don't have enough of these glycocalyx chains, then you're in trouble with being able to attract these signaling molecules. This is from Gerald Pollack's work. He has this schematic that's quite nice. He does simulations of these glycocalyx with special molecules, and he has shown that a battery is created. So here's the water, which would be in the blood, positively charged, because this is negatively charged here. It's pushing out these protons along the edge. And this is a cross section here where you can see around the area in here. Endothelium would be on this side here. This is the blood, the vascular sulfated glycosaminoglycans here, and then the blood is here. And then you have this battery that you create through this glycocalyx, and that is energizing the cells. It's like it's electricity supply for the cell. And here's another picture from some of his work. There's like this hexamer structure of this gelled water, and the protons are collecting along the inside here at the interface between the exclusion zone and the bulk water. So this is along the edge of the jello, you could say, is where the protons collect. And then you have this negative charge inside. And there's this quote: the surface energy of water is a non-metallic metabolic fuel source for the molecular motors of our body. That's quite a beautiful quote. So now glyphosate, a pervasive herbicide, is now the most used agricultural chemical ever. It's increased 50-fold since 1996. That's when the GMO glyphosate-resistant crops were first introduced. Today, 50 times more glyphosate is allowed by the EPA on corn grain than it was in 1996. Half of the American farmers' fields have weeds that are resistant to glyphosate. Now they're putting in additional toxic herbicides into the mix, adding, for example, 2,4-D. plus glyphosate. So this can handle the weeds that are resistant to glyphosate, making the situation even worse as far as our exposures. So the EPA claims that glyphosate is non-toxic, a wonderful herbicide because it kills all plants except those that have been engineered to resist it, but it's harmless to humans. It's not true. Our gut bacteria are impacted by glyphosate in a major way because they have the pathway that glyphosate disrupts, which is the shikimate biological pathway. And we depend on our gut microbes for so many things. And we're realizing more and more just how many things we depend on them to really keep us healthy, certainly supplying us with essential amino acids, which are produced through that pathway, and with many other nutrients such as vitamins and short-chain fatty acids. Other ingredients in Roundup are also toxic, and they increase the toxic effects of glyphosate. Glyphosate's insidious. Its effects are slow. It's a slow kill. And so if you study too short a time, you don't see the damage. 3 successful lawsuits claiming that glyphosate caused non-Hodgkin's lymphoma are bringing public awareness to glyphosate's toxicity. And now we have lots and lots of more lawsuits on top of those 3. Just my book, Toxic Legacy, How the Weed Killer Glyphosate is Destroying Our Health and the Environment, published in 2021. It presents extensive data. There's a huge number of references with each chapter, and it shows how glyphosate interferes with sulfate homeostasis, and it argues that glyphosate is insidiously cumulatively toxic through its diabolical insertion into proteins by mistake in place of the coding amino acid. This, I think, is central. Glycine substitution in the protein is, I think, essential for why glyphosate can cause so many diseases. It's really quite remarkable. And this book was selected by Kirkus Reviews as one of the top 100 nonfiction books of 2021. So here's a paper from 2021 that just shows all the different ways in which glyphosate causes damage, organ damage, effects on reproduction. Both male and female reproduction are compromised by glyphosate. It's a carcinogen, for example, non-Hodgkin's lymphoma. It messes up the DNA. It distresses the mitochondria, and it causes inflammatory and immunomodulatory effects. So reactive oxygen species, pro-inflammatory cytokines. Glyphosate really causes a very broad range of damage to the body. And here's another paper from 2019, Gut Microbiota and Neurological Effects of Glyphosate. Here's a quote from this paper. In this work, we state a possible link between glyphosate-induced dysbiosis and cognitive and motor aggravations in neurodegenerative and neurodevelopmental pathologies such as autism. I think glyphosate is central to the epidemic that we're seeing in autism. Hence, we review the negative impact that glyphosate-induced dysbiosis may have on depression, anxiety, autism, Alzheimer's, and Parkinson's disease. That's quite a list. So here's some charts. We have lots and lots of charts of this sort that I have published together with colleagues in multiple papers that show this incredible increase in all of these diseases that are happening over time in step with the dramatic rise in the use of glyphosate. And most of these are concentrated on the time from the first decade of this century, where glyphosate usage skyrocketed and all of these conditions became much more common than they had been before. And here's a brand new paper from 2024 that showed experimentally that glyphosate induced basically Alzheimer's disease in mice. They were exposed, they waited 6 months, and then they looked, and they found all of this evidence, various kinds of evidence that are symptomatic of Alzheimer's disease. So this is quite disturbing. And of course, Alzheimer's is one of the conditions that's going up dramatically. Now, this is crucial. Glyphosate impairs the NADPH pathways. I mentioned NADH is a major carrier of the depleted proton, and it's derived from tryptophan, which is one of the outputs of the shikimate pathway. So glyphosate is going to disrupt the supply of tryptophan. That's going to be one problem. But also it inhibits multiple enzymes that are involved with NADH, and in particular NADH dehydrogenase, which is the enzyme that actually delivers the H, the depleted H, to the ATPase pumps. Very, very crucial, this NADH dehydrogenase inside the mitochondria. It also inhibits this one, glucose-6-phosphate dehydrogenase. That's one that restores NADPH from NADP+. So that's also central for providing a low deuterium hydrogen to NAD. And then glyphosate inhibits succinate dehydrogenase. That's the only enzyme that participates in both the citric acid cycle and oxidative phosphorylation in the mitochondria. And a deficiency in that enzyme is linked to many cancers. So glyphosate substitution for glycine during protein synthesis, I kind of drew the schematic of a DNA strand. It has the DNA code. It breaks down into 3-letter units, and each one represents a particular amino acid. This is how proteins are made. And so here are our proteins holding hands like paper dolls. And what happens is glyphosate substitutes for glycine and messes things up. When the protein is being assembled, you end up with glyphosate instead of glycine. So here's sort of a protein being assembled. Here's your glyphosate molecule. The code for glycine, GGC, is coming in, is looking for glycine. It finds glyphosate because it has an exact match to the glycine piece that has to fit into the socket. So it goes in there and it has this extra stuff stuck on its nitrogen atom that messes things up. That's the big problem. So here we have a protein wrapping, a sequence of amino acids in a protein wrapping around an active site. The active site needs to fit the substrate, but if you put glyphosate instead of glycine here, you've got this extra stuff. There's your glycine piece, and there's extra stuff that's going to mess up the substrate. It can't get in because there's no room anymore because glyphosate has occupied that space. And that, I think, is really crucial, particularly for proteins that bind phosphate, and we'll get to that in a moment. So this explains how glyphosate disrupts EPSP synthase in the shikimate pathway. Multiple bacteria have developed resistance by replacing that glycine with alanine. And that's the basis for the GMO Roundup Ready crops. In other words, once you have alanine instead of glycine, glyphosate no longer fits into the socket, and you no longer have the problem. And then the glyphosate doesn't work at all. And so this is showing the mutant with the alanine versus the normal form. And the normal form, the glyphosate, as glyphosate concentration goes up, it completely clobbers the enzyme. This one is completely untouched by it. So it's quite a remarkable difference. And only glyphosate works. There's a paper about more than 1,000 different analogs of glyphosate that were produced and tested, and none of them worked. The only one that worked was glyphosate. And that's because none of them were amino acid analogs of glycine. That's what you need in order for glyphosate to work. So here's a Monsanto study from 1989. It was an unpublished study, but Anthony Samsel, my colleague, got a hold of it. And it showed, they looked at bluegill sunfish. They gave them carbon-14 labeled glyphosate, and then they looked for radiolabel in the tissues. They found radiolabel, but when they tested for glyphosate, they came up short. Only 20% could be identified as glyphosate. So then they got the idea of breaking down the proteins into individual amino acids. Once they freed up the glyphosate from the protein, then it could be detected. And they got up to 80% yield. 70% yield after they did that. And so they said perhaps the glyphosate was tightly associated with or incorporated into the protein. This is what I'm saying, incorporated into. So they were suggesting that back in 1989. And so Anthony and I wrote a paper in 2016 where we looked at all the different proteins that would be likely to be affected by this problem. And we identified a whole bunch of different diseases that we could see were going up dramatically in step with glyphosate, and that we could see could be caused by damage to certain proteins that have a specific pattern that matches what I call a glyphosate susceptibility motif. And so it's a glycine molecule that has an extra methylphosphonate. That's this thing over here. This is phosphate. Methylphosphonate looks a whole lot like phosphate, so it gets in the way of phosphate. And so it kills the weeds by suppressing EPSP synthase, and it blocks the binding of EPSP synthase to a phosphate in PEP. And that binding site has a highly conserved glycine residue. And when that glycine gets substituted, the enzyme is safe from glyphosate. So this is extremely strong evidence that this is what's happening. So many enzymes that glyphosate's been shown to suppress contain this motif, a glyphosate susceptibility motif, which I define as a place where the enzyme binds phosphate at a site where there's at least one, and sometimes 3 or 4 highly conserved glycine residues. So FAD binding in dehydrogenases is extremely important because dehydrogenases take hydrogen off of proteins and often off of molecules and often deliver them to FAD, to NAD to make NADH via FAD as an intermediate. And so they play a major role in supplying depleted protons to the mitochondrial pumps. Many of them are flavoproteins. Flavoproteins have this ability to select hydrogen over deuterium. They bind this FAD at a site with at least one highly conserved glycine, and this facilitates a transfer from the molecule to NAD. And so the FAD binding site has this region where it contains a highly conserved glycine aspartate pair, and that if there's a mutation in that glycine in that pair to alanine in a Bacillus NADH dehydrogenase, it causes a complete loss of activity. And that's because it can't bind to FAD. So putting glyphosate there is like creating a mutation like this, and that would cause the enzyme to be dead in the water. And so this also, an important feature is that the amino acids next door to the glyphosate need to be small so that there's room for its methylphosphonate unit. So here's these molecules, NAD and FAD. They're very, very important in biology. And they've got a couple of nucleotides that are tied together by these two phosphates. And this is where glyphosate causes trouble because it puts this methylphosphonate in the way and prevents these phosphates from binding. So now my first disease topic is colon cancer, colon cancer, colitis and colon cancer. And so this gets into sulfate because sulfate homeostasis involves 3 critical enzymes, all of which are going to be impacted by glyphosate. Sulfite oxidase, which converts sulfite to sulfate, phosphodienosyl phosphosulfate, PAPS, PAPS is the universal sulfate donor, and it's synthesized by PAPS synthase, which takes sulfate and ATP and puts them together to make PAPS. And then there's also sulfotransferases, which take a sulfate off of one molecule and put it onto another, extremely important for many, for the management of the sulfate system. All of these are expected to be disrupted by glyphosate because they all have very, very fine glyphosate susceptibility motifs. And so decreased decreased activity of PAPS synthase is associated with colon cancer. In a mouse model of colitis, PAPS synthase deficiency was associated with inflammation in the gut and the leaky gut barrier. And then glyphosate usage on core crops over time correlates strongly with inflammatory bowel disease. And so this is a detailed picture talking about a PAPS synthase molecule. It had talking about a sequence, GLSGAGKT. This has got 3 Gs. Those are glycines. And that's in a critical place in that protein. And they showed experimentally that when you substitute these 2 glycines with to alanine, just a small change, you clobbered its activity on both of it. It has these 2 different things that it does, and both of them got knocked way down with the substitutions of these glycines by other amino acids. So glyphosate would be expected to do the same thing. And then sulfite oxidase and sulfotransferases, there's molybdenum, which is a +2 cation. Glyphosate chelates all of these +2 cations, which would make the molybdenum unavailable. That's an important cofactor. And also the glycine residue at 473 is essential. If our aspartate is a very good model of glyphosate because they have similar sort of shapes and negative charge, that destroys the enzyme activity. So you would expect glyphosate to also destroy it. And then in impairment in the ability to bind sulfite and a 5-fold reduction in its activity. And so defective sulfite oxidase leads to severe birth defects and neurological problems. And then the sulfotransferases have this GXX-GXXK motif, which is required for binding to PAPS. So, and this is just showing you all these different versions of sulfotransferases that have this pattern of these 2 glycines that are going to get in trouble because of glyphosate substitution in various different animals and different versions of the enzyme. They are absolutely crucial for taking the sulfate off of PAPS and putting it onto all kinds of important molecules, the steroids like cholesterol and the sex hormones, vitamin D, the glycosaminoglycans, the chondroitin sulfate, the heparin sulfate, polyphenols and aromatics, these various beneficial nutrients, they need to be sulfated in order to get transported. And then the neurotransmitters, all the neurotransmitters depend upon sulfation for transport. So when you aren't able to add these sulfates, you get in trouble with being able to distribute these important molecules throughout the body. This is a paper on intestinal sulfation being essential for colitis and colonic carcinogenesis. So colon cancer is the 3rd most common cancer and the 3rd leading cause of death due to cancer. PAPS synthase expression is decreased in both mice and humans with colon cancer and is correlated with higher mortality and is associated with a reduced intestinal sulfomucin content and deficiency in the formation of bile acid sulfates. And here's a mouse model of colitis with the mice had this deficiency in this very important enzyme. And they had a leaky gut barrier and an acute inflammatory response. Leaky gut barrier has been shown to be connected to glyphosate. So you can see the deficient mice. You have this impaired barrier. These are the sulfated versions, and these are not sulfated. It makes the leaky barrier, and then things get across and cause all this inflammatory response. So this is a very common problem with many people in various diseases today in the gut that I think are caused by this problem of glyphosate disrupting the sulfate system. And here's a chart of the correlation between inflammatory bowel disease and glyphosate usage on core crops. It's from one of our papers. published in 2014. Histamine intolerance is my next topic. The big picture: mast cells release histamines in response to allergens. Histamines are metabolized primarily by histaminase, an important enzyme in the gut. Histamine sensitivity results when histaminase is defective, when it's suppressed, and a large number of uncomfortable symptoms arise from histamine intolerance. Glyphosate likely suppresses histaminase. So my hypothesis is that mast cell activation syndrome is induced when the gut lumen contains too much deuterium due to insufficient sulfate in the sulfomucins. And also another hypothesis is that the metabolites of histamine can trap deuterium and permanently sequester it. This is something that I'm playing with lately. Very, very interesting biophysical phenomena going on with certain special molecules. So here's a doctor who had a statement about mast cell activation syndrome, that there's an epidemic, millions of Americans suffer from multiple diagnoses, and they're put on an array of medications to try to stabilize their condition. They often don't improve. Their health continues to decline, and they're sometimes told their symptoms are psychosomatic, but the root cause is, in fact, this doctor argues, is mast cell activation. So this is something that has become really an epidemic in recent times, and that she says a unifying condition could explain all their symptoms, and that would be this problem with histamine intolerance. And so here's an example of from a paper that shows all these different symptoms you develop when you have histamine intolerance. Is quite nasty, with runny nose and asthma, diarrhea, so issues with the gut, arrhythmias, hypotension, menstrual cycle disorders, flushing, irregular circadian rhythm, all this headache and nausea and vertigo. A lot of different symptoms that are going to make you feel unwell because of histamine intolerance. So histamine, and I can't see this, YGHGGGWG, that is a amazing motif in histaminase that has a fantastic glyphosate susceptibility. It's really perfect. You can see all these glycines, 1, 2, 3, 4, 5 glycines in this motif. And this is there's a wonderful tool that allows you to put these images together, where you have this sort of description of all the different proteins averaged together that have this motif. And here, this is where they bind to FAD. And you can see here these glycines around this binding site right in here to the FAD phosphates. The P is the phosphate. So you have this FADP binding right in here at this place where there are these glycines. Here's your FAD with its double phosphate. So what happens is this can't work because glyphosate is jamming up the place where the FAD is supposed to fit. And so histaminase is diamine oxidase, which is also histaminase, is found in high concentrations in the intestinal mucosa. And they are the primary source of what shows up in the plasma, the same molecule, histaminase. A decrease in this histaminase reflects an increase in mucosal damage. And so circulating levels can serve as a marker of mucosal damage. A deficiency in histaminase is a primary cause of histamine intolerance. Heparin infusion increases expression of DAO. Heparin is the most highly sulfated molecule in the body. So again, there's not enough heparin sulfate lining the gut, which is causing inadequate gelling, which is causing too much deuterium in the body, in the gut lumen, I suspect, and that's causing these problems. The ingestion of histamine-rich food or alcohol or drugs that release histamine or block DAO may provoke diarrhea, headache, various nose problems, asthma, hypotension, arrhythmia, urticaria, pruritus, flushing, other conditions. So huge number of symptoms going along with histamine intolerance. This is a really interesting paper here that showed how you could look at the sewer system to figure out whether the community being served by that sewer system have a problem with histamine. And so they found a high correlation between the use of this Aleve and the weight in the wastewater and the amount of MIAA, which is a metabolite of histamine that I will say more about in a moment, a very interesting metabolite of histamine. And so this allows them to assess the disease risk in a population by just looking at the sewer system. I think this will be a powerful way to identify communities where there's a generic problem of some sort, probably excess glyphosate exposure, that's causing them to all have this problem. So this is MIAA, methyl imidazole acetic acid, a very interesting molecule derived from histidine. And I want to point out this H right here because this carbon atom is extremely interesting. In this ring, it easily picks up deuterium from the water due to its nearby nitrogen atoms and due to this ring. So there's a very special place for a carbon atom to be which allows it to actually exchange hydrogen with deuterium. And the really cool thing is that when it does so, it won't let go. And so this C can become CD by reacting with the water, with the deuterium in the water, and then it becomes permanently trapped on this molecule. And this one, MIA, is especially good at doing this, at trapping a deuterium on its carbon atom. I am suspecting that it is being produced in order to clean up the deuterium that's in the water, because there's too much, and there's too much because the sulfomucins are sick. So this is a very interesting paper that showed various kinds of molecules that contained this imidazole ring, and what kind of, as a function of pH, the imidazole ring becomes much more able to pick up deuterium when the pH is high. So if the pH is elevated, then there's a much greater chance for these molecules to pick up deuterium. And this is a particular protein that has a peptide sequence that has a histidine that they're looking at right here. So this is just one molecule, but this one is several different versions of molecules that contain this imidazole ring. And IPA, this is the one that we were just talking about, imidazole propionate. This is imidazole propionate, which is made by bacteria that has a really good job, a very good ability to trap deuterium. So I'm thinking that imidazole propionate is actually being produced by the microbes to trap deuterium. And so I can remove the deuterium landmines. That's this imidazole propionate. And so this is actually produced by microbes. It's produced under conditions when the pH is elevated, which is those are the conditions in which it can trap deuterium. It inhibits insulin signaling by activating the mTOR pathway. So I think it's a signaling molecule that recognizes that there's a deuterium problem and it wants to fix it. So this is an explanation of the gut disruption leading to IPA overproduction. This paper talks about the enzyme that produces IPA is active at neutral pH. The proximal colon has an acidic pH. Increases in pH possibly resulting from decreased production of short-chain fatty acids and increased protein fermentation. These two things can cause the pH to rise, which can increase the production of this imidazole propionate. Glyphosate causes an increase in the pH, and this greatly reduces the production of short-chain fatty acids by acid-loving microbes. I talk about that in my book, in my chapter on the gut. And it also increases the production of ammonia, which raises the pH from protein fermentation. So in other words, glyphosate causes these two problems, and these two problems cause the production of this IPA, which is linked to diabetes. And so I think it's really, really important that the gut microbes are supplying short-chain fatty acids to the gut, and in particular butyrate. Butyrate is kind of food of the gods as far as the colonocytes are concerned. This is a schematic of a colonocyte. Here's a mitochondrion inside the colonocyte. The butyrate comes in and goes into the TCA cycle in the mitochondria, and it produces deuterium-depleted water in the process of metabolizing the butyrate. And the butyrate is extremely low in deuterium because it comes from that hydrogen gas that was produced by the microbes. So these are dietary fiber, gets degraded by these primary degraders, and then secondary degraders produce the hydrogen gas, and then the hydrogen gas is combined with carbon dioxide to make acetate, propionate, and butyrate. These are the short-chain fatty acids. Butyrate in particular is what the colonocytes love, and it has a low deuterium, and it keeps them healthy. And that is really, really important. When they don't get enough butyrate, then their mitochondria get sick. So to recapitulate, mast cell activation syndrome was first recognized as a disease in 2007. It's associated with excess inflammation, allergic reactions. They release abundant histamines into the gut lumen. Histamines act as a signaling molecule to induce all these problems. Histaminase metabolizes histamines into metabolic products, so removes the histamines, and that those products may be able to trap deuterium and remove it from the gut. Suppressed histaminase causes histamine intolerance. Histaminase has a very strong glyphosate susceptibility motif. Gut microbes metabolize histidine into imidazole propionate, which is a very good molecule for trapping deuterium. And so it's quite possible that this imidazole propionate is able to help to clear the deuterium from the gut. Glyphosate induces an environment in the gut that is conducive to IPA-producing bacteria. Impaired fatty acid metabolism, my next topic. Why so many picky eaters? An observation is that many children in America, and especially autistic kids, are very picky eaters. They refuse a whole lot of foods, and they will eat only a few things. And often those few things are not good food. They're processed, heavily processed foods. So are there enzymes that could be suppressed by glyphosate to cause this condition? Now, this is a paper that I published together with Judy Hoy and Nancy Swanson back in 2015. Again, we were looking at correlations. We found a very strong correlation between newborn metabolic disorders over time compared to the rise in glyphosate usage on core crops. So we included feeding problems, poor weight gain, slow fetal growth, congenital anomalies in the gut, and fetal malnutrition. And so these are all happening at higher rates in more recent years because of the excess exposure to glyphosate. So I've identified this protein, long-chain 3-hydroxyacyl coenzyme A dehydrogenase, LCHAD deficiency. This is a dehydrogenase, so it has this property of being susceptible to glyphosate's mischief, and it plays an essential role in the metabolism of long-chain fatty acids. And when you have deficiencies in these genetic deficiencies, you have severe disease with feeding all these kinds of things that are showing up in association with glyphosate, increasing over time in association with glyphosate, all these different kinds of problems. Neonates that have a severe phenotype present within a few days of birth with low blood sugar, enlarged liver, brain dysfunction, and cardiomyopathy. So very, very critical enzyme in the body. Glyphosate has been shown to suppress dehydrogenase enzymes in many studies on human cells, E. coli, and soil bacteria. So it's very clear that dehydrogenases as a class are suppressed by glyphosate, and they are flavoproteins. They have that whole process that they're able to select hydrogen over deuterium. So they're very important in pulling hydrogen off of organic molecules and giving it to the mitochondria. Patients with an LCHAD deficiency appear to have a specific cognitive pattern, presenting an intellectual disability and specific autistic deficiencies. So I think that this enzyme is part of the problem with glyphosate and linking it to autism, and weaknesses in auditive verbal memory and adaptive and executive functions. So this is this lovely tool that I use to figure out whether something has a classic glyphosate susceptibility motifs. You can just throw the sequence for the protein into the input box of this and push go, and you get this lovely result, which is pictures like this. It's really fun to use this, and I've been able to find a lot of proteins this way that would be expected to have a glyphosate susceptibility motif. And so this one, LCHAD, is amazing because it has all of these. There's 5 different hits, and I haven't found another protein that is so spectacular in terms of glycines, containing glycines at places where there's important things happening. And these are binding phosphate, in particular to bind phosphate. So these are all going to be problematic. And so this, I can't imagine that this enzyme is not affected. I can't imagine it's not affected by glyphosate. And so here's this this motif. You can see there's all these FADs and NADs and ATPs. All of these things bind to this place where there are these 3 glycines at this in this area, and you can see all these boxes are where they bind. And so you're going to have enormous difficulties with this enzyme if you've got glyphosate in the mix. It suppresses EPSP synthase, which we've said before, at a site that binds phosphate where glycine is highly conserved. So all of this is going to be problematic. LCHAD is essential for fatty acid oxidation. Here's a piece of the activity that goes on to produce with LCHAD. LCHAD is right in here. Is a sequence that actually pops off one of these acetyl coenzyme A's off of the fatty acid and shortens the fatty acid. This is how the fatty acids get metabolized. This gets sent off to make AGP, and they're naturally low in deuterium. So each round through this pathway peels off 2 carbons from the fatty acid chain to release the acetyl coenzyme A. LCHAD deficiency blocks this oxidation in the mitochondria. So that's a huge problem. You can see here's the H2. This is going to be deuterium depleted protons over here, deuterium depleted H, and this is going to also be deuterium depleted. So this is a paper on glyphosate. Glyphosate excretion is associated with steatohepatitis and advanced liver fibrosis in patients with fatty liver disease. And so this is non-alcoholic fatty liver disease. It's the most common chronic liver disease in developed countries. It's becoming more and more common over time, and it can progress to a more severe form, which is non-alcoholic steatohepatitis, NASH. It increases the risk to cirrhosis and liver cancer. Rats develop fatty liver disease after low-dose exposure to glyphosate. And so this is an important paper here. We report that glyphosate excretion is significantly higher in patients with NASH compared to patients without NASH. In addition, we also report a significant dose-dependent increase of glyphosate exposure with increase in fibrosis. In other words, the worst cases of NASH have the most, the lesser NASH has less, and no NASH has the least. And this is significant differences among all three by just looking at glyphosate in the urine. I found that quite a remarkable thing. So LCHAT is essential for metabolizing long-chain fatty acids. Genetic defects are associated with severe developmental problems like autism. LCHAT has 4 sequences containing glycine where the protein binds phosphate, so that's a very strong glyphosate susceptibility feature. Glyphosate has been shown experimentally to disrupt fatty acid metabolism, and that could well be because of this deficiency in LCHAT. NAFLD is an epidemic today and is associated with high levels of glyphosate in the urine. Fatty acids are low deuterium nutrients, so impaired metabolism leads to excessive mitochondrial deuterium and mitochondrial dysfunction. So in summary, deuterium is a natural element with biophysical and biochemical properties distinctly different from hydrogen. Deuterium management in the body involves sequestering deuterium in gelled water lining the gut and the vascular barrier. Depleted protons are released into the gut lumen and the circulating blood. Glyphosate suppresses many dehydrogenases and enzymes involved in sulfation, impairing gel formation and derailing deuterium homeostasis. Colitis and colon cancer are associated with defective sulfomucins. And my hypothesis is that histamine release may be a mechanism to diffuse deuterium landmines in the gut. Peer-reviewed papers show that lipid oxidation in mitochondria is suppressed by glyphosate, and lipids are low deuterium nutrients. Okay, and we are done. Stop the share. Questions?
A million.
Loads.
But yeah, so how shall we start? Rachel, do you want to start?
Yeah, I would really love to start if that's okay, because I'd like to kind of bring this back to clinical practice, Stephanie, because personally, as a nutritionist, I see so many people in clinic with all of the things that you've talked about: mast cell activation syndrome, histamine intolerance, post-orthostatic tachycardia syndrome, hypermobility. And what you've just presented is such a clear picture for me that I just felt that I needed to kind of get your clarification on how I've understood it. So it's glyphosate that's the big kind of high level top problem here. It's kind of having a detrimental effect on the sulfate metabolism pathways, the tryptophan pathways, the histamine pathways, and collectively this is contributing to all of the symptoms that we're seeing potentially in this long COVID type presentation. So one of the questions that I had was that in general nutrition clinical practice is that we see a lot of people with, say, small intestinal bacterial overgrowth, and people are testing this and showing high levels of hydrogen sulfide gases and methane gases coming out through the breath. Now, I've intuitively thought we don't want to be getting rid of that, because I feel in this presentation it's actually having a beneficial effect. Is this because these gases are depleting deuterium and they're trying to salvage these pathways? Could you just kind of
Yes,
it's
pretty simple, actually. It's pretty simple because the hydrogen gas is where you start. So there's these microbes that are able to, they have an enzyme called hydrogenase that produces hydrogen gas, and it's 80% down on deuterium. So it's a wonderful resource if you can use it to make organic matter. And there are other microbes that take the carbon dioxide, the gas, and the hydrogen gas, and they make methane, and other ones make acetate, acetate and methane. And both of those are supposed to, methane is still a gas, and the methane then goes through a sequence becoming methanol and formaldehyde, and then finally formate. And formate actually, it gives its 2 hydrogens, which are severely depleted in deuterium. It gives them to methyl groups through the methylene tetrahydrofolate is produced from the formate. So that methylene tetrahydrofolate has a very low deuterium in the methylene. And then the enzyme that adds one more high H to make methyl tetrahydrofolate, that methyl is going to be really low in deuterium. So I think the entire methylation pathways, I have some slides on that, but I didn't include them here today. Yeah. The methylation pathways involve capturing that methyl and delivering it to various places where it's a source of a low deuterium nutrient. It finally gets metabolized in some place in the body. So that's just
Absolutely.
one example. But what happens is all of these dehydrogenases are suppressed by glyphosate. I think all of them as a class. There's a study on E. coli that found 12 different dehydrogenases that were suppressed by glyphosate. They all have this glyphosate susceptibility motif. They are flavoproteins. They're able to use hydrogen tunneling, proton tunneling to keep the deuterium out. They're really fancy. So if they see a version of the molecule that has a deuterium at the place where they want to yank off the hydrogen, they won't touch it. They just leave it aside.
Yeah.
So they're selecting for the ones that have the good hydrogen to make a product that's low in deuterium, and they are suppressed by glyphosate. So when you have the methane, it can't get in, it can't get back into the body as something you can save. And that's why you have the cows releasing lots of methane gas, and now they're killing all the cows. You know, what they should do is feed the cows an organic diet so they can get that methane into organic matter. You know, and so the gases are getting stuck because of the dehydrogenase. It's not working. The gases are sticking around, and then you get the bloating because they're not being removed by these enzymes that would normally turn them back into organic matter, like the short chain fatty acids.
Yeah, I get you. And with regards to the histamine pathway specifically, so when I've kind of broken down the actual whole pathway to how it gets metabolized through the urine, there is an imazidol ring on every single metabolite through the histamine detoxification pathways. Is it the imazidol ring specifically that's depleting deuterium as well? Have I understood
That's
that
what I
correctly?
think. I mean, it's really I've got a paper under review. I have a preprint in this with some in collaboration with Anthony. Pardon? That talks about various really fancy. It's really cool, actually. There's a small set of molecules that are able to trap deuterium, and one of those is these is lipids with bisallylic carbon. It's called bisallylic carbon atoms, for example, in DHA. DHA has 5 of those. And those bisallylic carbons are very reactive. So there's a problem with them. There's this reaction cascade that happens with lipid peroxidation. But in that process of lipid peroxidation, they pick up deuterium. And when they pick up deuterium, it actually resolves the inflammation. It's truly amazing. I'm working on a paper right now that goes into that. It's so fascinating. But then the histidine, also the imidazole ring, is able, I think, to trap deuterium, and especially like, for example, the imidazole ring in the imidazole propionate, which is produced by the microbes in association with diabetes. That's so interesting because I think the pH goes up, that compromises butyrate production. So the colonocytes get sick because they got too much deuterium. And that whole system sets up on, oh my God, we've got to do something about this kind of reaction among the gut microbes. Let's make some imidazole propionate, which only gets made if the pH is high enough, because that enzyme requires a high pH. So it's detecting the high pH, which it knows will be problematic. And the high pH is a consequence of other things that glyphosate does. Like it disrupts the breakdown of proteins in the upper gut. So the proteins make it down into the colon. And then in the colon, they get metabolized to ammonia, and the ammonia raises the pH, you know, and so then you can't make enough butyrate, and then you go down, you go south. It's just it's a really complicated process that ends up with disease.
And I see that there's a tie-in with UV light in amongst all of this as well, because that histidine, when it's exposed to UV light through the skin, can convert to uricanic acid. And again, this is another pathway into kind of making folate, or kind of metabolizing that folate pathway, but actually causing reactive oxygen species, which also play into this whole cascade as well. And I think that's something that I've been looking at, but also UV light tying into the sulfation pathways and cholesterol and female, or all of the hormones that are produced from that sulfation. Is that something that could potentially be used to support glyphosate detoxification? Do you think that could be a pathway that could work?
That could be interesting, whether the UV light could actually destroy the glyphosate. I think it's possible that it could. Certainly, you know, there's oxidizing agents can destroy glyphosate, chlorine dioxide, chlorine itself, and hydrogen peroxide, superoxide. These kinds of things can, you know, energetic oxygen can destroy glyphosate, and non-enzymatically. And so and I'm actually really glad that they use chlorine in purifying the water, because I think they're trying to keep the bacteria out, but they're also, I think, breaking down the glyphosate with the chlorine that they use in the water processing. which is
Yes.
fortunate because I think we would end up having a lot more glyphosate in our drinking water if they didn't do that.
Absolutely. And one last question. Sorry, Joachim. I know you got some questions too. Deuterium depleted water for drinking, Stephanie, is that something that, I mean, I'm imagining this could potentially help.
This is deuterium depleted water. I drink it
Yeah.
regularly. Every day I have one glass of 2/3 of regular water and 1/3, just 1/3 cup of a very low deuterium water that I buy online. From drinklightwater.com. It's called, is a organization where they sell it. Very expensive. It seems ridiculously expensive. It's like buying wine when you buy water, but I only use 1/3 cup a day. So that's kind of a maintenance dose. I think if I had cancer, I would drink a lot more.
Yeah.
And I mix it up with the regular water, so it feels like glacier water. So basically you can also get glacier water, and glacier water typically is low in deuterium as well.
Oh, fantastic. Thank you so much.
Mm-hmm.
Thank you, Rachel. Thank you, Stephanie. Look, let's zoom out a little bit now, because we have a lot of people in the audience that are doctors and medical practitioners, and they want to know what to do about it. And so if you look at these two, I mean, you and myself, and you more than myself, have looked at these two champions of causing a complete train wreck in the organism, that is SARS-CoV-2 spike protein and glyphosate.
Absolutely. They're both equally fascinating, actually. In terms of what they can do to biology, they're so amazing in their skills to destroy everything.
If you look now, we have listeners from all over the world, United States, Canada, but also South America, Africa, Asia, and Europe. And so you can probably state that the sickest populations are where the highest glyphosate use is also reported, or at least the highest glyphosate content.
Certainly.
Yeah,
certainly true for the US. We have miserable performance with COVID, much higher death rates than other countries. And we also have we're probably equal with Brazil in terms of the amount of glyphosate we use on our crops, comparable to Brazil.
And
which is the worst in the world,
So we have two, let's say, overlapping problems. One is in the United States, over 60% of the general population, including children, are already reportedly chronically ill and take medication for chronic illnesses. And now, on top of such a vulnerable population, you introduce an agent like SARS-CoV-2 and its spike protein. So no wonder that we see what we see now, almost starting to see a systemic problem in the general health of the population. I mean, we get data from insurance companies, we get data from other sources that clearly report 10 to 15% of the overall population already suffering on top of the chronic illnesses now with something we could call long COVID or post-vax syndrome. And so how to go about it? Because now you have these doctors facing patients that display maybe a chronic illness, but we know that both glyphosate and SARS-CoV-2 spike can introduce or fuel or trigger or cause cardiovascular, neurodegenerative, metabolic, intestinal disorders, and also lead to epigenetic dysregulations and cancer. Both the same and with many overlapping symptoms. So what would you recommend? I mean, I sent a list beforehand to Rachel also to work out a nutrition plan, because it's not only to detoxify. Detoxifying is one way, same like with the spike protein. If you detoxify glyphosate or spike protein from the organism, that is not the whole equation. It will not cure the patient. It will take out the cause of the disease, but you still have to do a lot in nutrition-wise, dietary supplement, lifestyle-wise, and also have to have some patient to correct these problems. And so now we are facing not only one side with all the chronic illnesses caused by glyphosate, we also have on top now the spike protein. And so how would you go about it if you were a practitioner, or you have to, where would you start?
Well, I think, getting rid of the source with glyphosate is to eat organic, eat certified organic food, have your water tested. If it is contaminated, add a filter, put in a reverse osmosis filter to get the glyphosate out. And so I think it's really important to eat certified organic, and of course also whole foods instead of processed foods. Avoid all the food that's in boxes, and cook your meals at home, and eat a lot of fiber. Fiber, it turns out to be important as a source for the butyrate. The butyrate is so important for the gut. I feel like In fact, butyrate protects from histamines. It suppresses I think it's the deacetylases of histone deacetylases get suppressed by butyrate, which is a way to protect from mast cell activation syndrome. And so, but butyrate has many properties and healthy, health-giving properties. So it's really important to try to get enough butyrate. Even you could take butyrate as a supplement or eat foods that contain butyrate, like butter. Butter is a good source of butyrate. Eating a high-fat diet is important. And I also think animal-based fats in particular are very important, but it needs to be like grass-fed beef, organic pork, these are hard to come by sometimes. Organic pork is very difficult to find, but bacon is a very healthy food if it's organic. And of course, seafood. Seafood is a really good source of taurine, and taurine is also a very interesting sulfur-containing molecule that I believe the gut microbes can convert into sulfate. So there's a sulfur deficiency problem, I think, in our society with the foods that we eat. Our foods are depleted in sulfate in sulfur, in part because of glyphosate. And Don Huber has shown that glyphosate severely disrupts the uptake of sulfur into plants, so that the plants that are supposed to contain sulfur don't contain enough. But you can eat sulfur-containing plant-based foods like garlic, onions, cruciferous vegetables. Those are all really good. I eat a lot of those in my household because they're a really good source of sulfur. And then you have the animal-based source of sulfur, which is like eggs and seafood, clams and crabs and lobster. All of those are very nutritious. They have a lot of micronutrients. So you want to sort of eat foods that are dense in micronutrients, and you want to eat a high-fat diet and a high-fiber diet is kind of the profile that I would recommend. And fermented foods as well. Fermented foods are really good.
We could look at two things. So a recommendation would be good if between you, me, and Rachel, we would work out, first of all, which kind of food stuff is most contaminated, so that people that can't afford all organic at least stay away from very contaminated food stuff that contains a lot of glyphosate, like cane sugar or
legumes. Yeah, legumes. I think
legumes are really high. That's the highest in Canada. The highest numbers were found in legumes, and so hummus, for example.
And then seek a nutrition that will, like for example, also maybe supplementation of glycine to push out the humic acid that we develop that takes out that chelates glyphosate in the gut. And by the way, also spike proteins, so pretty nice combination.
That's really good.
We have a lot of tools, but we have to make them accessible to everybody that's listening here, so that they know, okay, stay away from these foods, seek those foods. For example, you have the fucoidons and some other algae products, like in wakame, the Japanese algae that you get with sushi. These are highly in sulfur. They are actually heparin analog kind of products, and they chelate spike, and they rebuild the glycocalyx. Maybe interesting for everybody listening why it's so important was Stephanie was saying today. We tried in Japan in our laboratory to get hamsters sick on spike protein. And we injected a very high dosage of a mRNA vaccine into the heart. And guess what? They were young and nothing happened. Nothing.
That's amazing. That must have been a disappointment.
No. I told Kevin, I said, you have to inject them LPS first, or give them a high glyphosate containing foods if you want to get them sick. And then we looked at the intricate details. So if you have an intact glycocalyx, there's even papers showing that not even lipid nanoparticles of mRNA vaccines can penetrate the endothelium if the glycocalyx is intact. So
so that's the problem, is the sulfur.
So
I think that, and because heparin sulfate deficiency in the brain has been linked with autism in both humans and mice. And they've even identified deficiencies in the enzymes that make the heparin sulfate, because the sulfation enzymes, which is the ones that are affected by glyphosate.
I was going so far to say that the glycocalyx determines if you become sick of long COVID or post-vax syndrome. That is one of the most determining factors from all. And the crazy thing is, once spike protein or SARS-CoV-2 virus enters into the endothelium, it immediately upregulates an enzyme called heparinase, and that will degrade glycocalyx and any heparin-containing compound. And that means it was like a bushfire. breaking up the glycocalyx to get to the endothelium, and that's when the process starts. And then from there comes another 100 cascades downstream, what will make you sick. And so your connection between glyphosate and the glycocalyx is essential. And another endorsement, I think, that people should avoid glyphosate-containing food, seek foods that counteract this. And so if DFW is interested, let Rachel and myself and you work out a list, what to stay away from, what to do, what kind of foods, what kind of supplements to take, and in order to get that problem somehow mitigated, because now we're not only having to deal with spike protein, we have to deal with glyphosate intoxication at the same time, because all the overlapping synergistic mechanisms are fueling each other. And that is
Exactly
in the making.
Yeah, there was I remember an article that was talking about the glycocalyx and the sulfation as being so important in COVID-19. It was actually an article about severe COVID being associated with impaired heparan sulfate proteoglycans.
But are there other questions, Raul? Are there questions from the listeners that we want to address also? Because I know Stephanie doesn't have all the time.
Joachim, I have a question to Dr. Stephanie. That how does the chlorine dioxide work on the glyphosate?
Yeah, well, it's one of the molecules, small molecules that are able to break it down non-enzymatically. Just because it's so reactive, it's an oxidation. It's an pro-oxidant as opposed to an antioxidant. So it can oxidize the glyphosate and break it down non-enzymatically.
Okay, that's great. That's great. I can see Kerry Rivera is here, and she's sending her special love to you. Kerry, would you like to say?
Wonderful to hear you, Kerry. Thank you.
Hi, Dr. Seneff. Thank you for being here. I'm great. How are you?
I'm very good, thanks.
Love to you both. Hi, Joaquin.
Okay.
Bye, guys.
Yeah, Kerry has done some very interesting work with chlorine dioxide and autism, and she's getting some great success stories. So I really appreciate that.
Correct. Thank you so much.
I think if you talk about chlorine dioxide, I think one of the biggest advantages is we see a very strong overgrowth in pathogenic bacteria, fungi, and other pathogens in general in the post-COVID post-COVID patients. And so chlorine dioxide will mitigate that. And there's a lot of mycotoxins, there's a lot of overgrowth in bacteria that you don't want to have. And so I think there's one of the biggest potentials where we can see that chlorine dioxide could do something useful apart from degrading spike proteins. But
yeah, I actually saw a documentary, a 1-hour documentary on chlorine dioxide. Was quite interesting, and they specifically talked about COVID patients who found significant improvement in long COVID with chlorine dioxide.
Yeah, we have to see. This has to
Thank
be.
you so much. Yeah.
Susan, you're working on that, so we will get data from you soon.
That is, I consider chlorine dioxide as one of the method in which you can have oxidation in hydration. So I always when I do it with my patients, I tell them that one is you're breathing oxygen, here you can drink oxygen.
Yeah, I know. Yeah,
but I think that we could ask one more time to Stephanie. Look, if you can bring down the glyphosate levels, you need a lot of glycine to push it out of the proteins that it already has been incorporated into, because then the body takes glyphosate instead of glycine, and then screwing up the protein completely.
So bone broth is a good choice for that too, because it has a lot of glycine. Bone broth is a very healthy food.
And bone broth also is a very good food that can restore the glycocalyx, by the way.
Exactly, yeah.
So why don't we make a list? Let's do that. A very simple list for all the doctors and practitioners and whoever's interested, what foods to take, which are taking care of the glyphosate problem. You have another big problem. Maybe you want to describe that very briefly. And I know that Rachel has been on the topic with me for a long time. Is all the neurotransmitter precursors like tryptophan, and the metabolization of that, because the same chain is heavily hit during post-COVID and post-vac conditions. And that is causing, let's say, the neurological symptoms and problems, at least to a big part. So maybe you want
Yeah,
to
I mean, I think tryptophan, tyrosine, and phenylalanine come out of that shikimate pathway, and the gut microbes make those molecules for the host. So when the microbes are being exposed to glyphosate, you get deficiencies in those very important amino acids. Those are coding amino acids, and they're also precursors to all the neurotransmitters, and also thyroid hormone, and the skin tanning agent, melanin, melatonin, serotonin, dopamine, adrenaline, melanin, thyroid hormone, they all come out of that shikimate pathway. So all of those are going to be in trouble if your microbes can't make enough of those, and you don't get enough from your food supply of those critical amino acids. And glyphosate suppresses the synthesis of those amino acids by the gut microbes, but glyphosate also suppresses the sulfation of the products. As the tryptophan is transported in the gut, through the blood with the sulfate attached to it. So if you can't add the sulfate to the tryptophan, you can't ship it out. So that's another problem with distribution of all of these. All of these neurotransmitters are sulfated in transit. Melatonin is sulfated. Serotonin is sulfated. They're all sulfated in transit. And that enzyme that adds the sulfate is probably compromised by glyphosate. So, and of course, the entire sulfate chain is compromised by glyphosate.
Well, this will be very, very valuable
Yeah.
for everybody in this industry and in the field to understand that at least in the populations that are heavily exposed to glyphosate. I don't know how it is, for example, in India. I wouldn't know how it is in other parts of South America, but I know in Argentina it's very, very high. And I think also in Brazil, so that the doctors and practitioners understand all that, because we have two big problems. One problem is, of course, that many long COVID post-vac conditions are not recognized because they hide behind the mask of a chronic illness. So you see a cardiovascular problem, you see thrombosis, you see some metabolic disorders, diabetes too. As a doctor, you don't suspect that it is caused by the spike protein. You think it's just a normal case of a chronic illness. That's one thing. And then deeper than that, you have the glyphosate already preparing the ground and disrupting the organism before the spike even hit. So we have to take both of these mechanisms and processes into account if we want to provide solutions, because the reason why we do this research is somehow to help and to bring out solutions. So let us do this kind of, we can make it as a paper even or whatever, so that everybody can read, okay, stay away from this, do this, rebuild the gut. The gut is the center stage of all, also for long COVID and post-vac. I mean, you see that with Bronja's finding now that the bacteria is being infected by the virus, and that
that's wild.
And we just for everybody's information, we are finding now we have a new diagnostic method, and we find bacterial involvement and toxic peptides produced by that process in pretty much all patients.
Wow.
At least the blood analysis that we have, it's not hundreds yet, but whatever you look at, it's immediately high, very high in toxic peptides. So you have already a very disturbed gut biome through the glyphosate, and now you introduce SARS-CoV-2 and its spike protein into the bacteria and into the intestines, where it will synergistically have a massacre on the bacteria, decrease butyrate and short-chain fatty acid producing bacteria, cause leaky gut on top of what glyphosate also does, endothelial inflammation, and then start from there to get out into the bloodstream and do the rest. So the gut is becoming more and more interesting now also in the sense of gut brain or the gut brain axis.
Yes, that's so interesting.
Yeah. So let's get together. I'll send an email to both of you, and we create such a list as a takeaway today. Is your presentation available for everybody that we can look at it?
It can be for sure. Should I send it to someone and you can post it somewhere, or I can put it up on my MIT website,
or maybe can you tell us can we put your website up so that people can just go to your website even? It's easier.
Okay, but it's not there yet, but I can put it there. So I don't know. I could maybe
I have a question.
Okay.
Dr. Seneff, congratulations for a great lecture.
Oh, thank you.
I am a functional doctor from Brazil, and I know your work since 2014, when you warned about the important increase in autism incidence, and now we are almost getting there, those numbers.
Oh, it's
at least
sad,
in
isn't
Brazil.
it?
And
That's
I would
unfortunate.
like to ask, about ozone therapy. We have used ozone medicine therapy with great success in many chronic diseases. And I would
Interesting.
like if the ozonized water could help to detoxify glyphosate from water.
If the ozone could remove the glyphosate from the water, right? I think so, because I think ozone breaks down glyphosate. And that might be why it's so beneficial.
Yes, we have been using it for medical therapies and also to help to clean our food. In Brazil, as you said, we have, at least in my state, Paraná, it's a great, we have a great production of agricultural products, and we have
Right.
4 times the amount of glyphosate than other parts.
Oh,
So
you are
we
one of
have
the
been
worst
using
offenders within
ozone
Brazil,
water.
right? Your county is one of the worst.
Yes.
Yeah, that's very sad. Yes.
Okay, can I ask Jenny
So-
Gazilia to ask her question?
Okay. Hi, Stephanie.
Hi.
Hi.
How are
Well
you?
done. Wonderful speech.
Thank you.
When you were speaking about both of you, Joachim and Stephanie, about the spike protein of COVID spike protein, I see the glyphosate. We've spoken, Stephanie and I, we've spoken about this before. The fact that it's a chelator, it's a very strong chelator. From what I see, Joachim, you just mentioned the fact that there is the sulfate pathway. So Stephanie, I'm going to ask you a question on this. The sulfate pathway is actually very, very needy of copper. And when we look at the and then when we look at the deficiency symptoms of copper, where it breaks down the cell signaling pathway, which we see a lot in neurodegenerative diseases, cancer, so we're including autism, Parkinson's, and all the rest of them, but also the motor issues. And then we're seeing exactly the same symptoms arising. Stephanie, you've done so many papers on this, from glyphosate toxicity. The overlap seems so identical. And I know that the FDA actually published in 2021 a press release stating that COVID-19 could be killed 99.9% by certain copper alloys.
Oh, wow.
Yeah. What do you know of any connection between glyphosate, copper, and the spike protein? Have you come across that?
Well, certainly glyphosate and copper. Copper is one of the minerals that glyphosate chelates, and so it's going to mess up the body's natural methods for transporting copper. And I'm trying to think of the name of the protein. There's a protein that carries copper and also influences iron uptake from the gut.
Ceruloplasmin.
Thank you. That's the one, ceruloplasmin. I actually looked at that one, and it has a glycine residue at every place where it binds copper. And so I'm suspecting that glyphosate could be substituting for glycine in that ceruloplasmin and disrupting its ability to bind copper, which is then going to mess up the entire system that delivers copper to the entire body.
Yeah.
So I think that's a very interesting thought with the ceruloplasmin being affected by the glyphosate.
Okay. I find it also interesting when we speak, just one more thing, on histamine intolerance. Histamine intolerance, I know you would have known, diamine oxidase is actually the regulator of histamine, which is
Yes.
copper dependent. So when you remove the copper, the regulator of histamine goes out the window, and then histamine levels build up. The overlap is so clear. I've written a paper on it. I'd be happy to send it to anyone who wants to read it. But, I love your work, Stephanie. You know that. I've told you that for years. But, thank you so much, all of you, because it was really interesting.
I have to say that copper and histidine interact very much so in many proteins, and especially in amyloidogenic proteins. I've been researching that topic lately. I suspect that the copper facilitates the trapping. This is a smoking pot kind of hypothesis at this point, but it's something I'm working on right now, is the idea that the copper, because copper is carries electric fields very, very well. So copper may facilitate the uptake of deuterium by the histidine, by the imidazole ring in the histidine in certain proteins, and that is an important role that they play in trapping the deuterium in that histidine molecule. I'm suspecting. I'm working very hard right now on neurodegenerative diseases and misfolded proteins to try to understand the role of deuterium in all of that.
Thank you.
Maybe one field that we have not touched yet is the mitochondria, because I got the information from a laboratory here in Germany from Professor Bridget that she's a mitochondria expert, and she has a mitochondrial lab. She says all patients, without exception, long COVID post-vac, are showing complete destroyed mitochondria.
Oh, wow. Oh, my. That's amazing.
So that is another thing another topic that you touched today on NAD, NADH, and ATP and the pump, and so if let's say if the glycocalyx is one of the main factors to keep that EZ zone and to keep that gel-like water trapped and everything so we have to go and see that we have to do this at the same time. So again, that on top of the list with the glyphosate and the foods would be another big thing for a separate stream to talk about mitochondrial health, because I think, Rachel, you're in that field very well positioned, and we have to see, because the mitochondria is if they are not functioning well, then we have no chance to maintain our health. It's impossible. So that is one of the most, next to the gut and some of these issues, one of the biggest fields that we have to see how to revamp that. It's not only about taking out spike protein, it's about all the other measures we have to take now to bring people back into something that resembles a healthy condition. So maybe we can add that as well. So I'll put that on the list between us. I might write an email in there. Raul, did you receive? I sent you by WhatsApp Stephanie's link for her website.
Yeah, and I don't have those slides up there now, but
you have a website. It's called stephaniesenef.net. Is that okay?
Yeah, I have that too. So I could let me try to make a note to put it up on both of those sites. And it'll be glycocalyx.pptx. You can look for that on that site when I get it up. I'll have a header for it. Post it. Just make a note here. I can put it on both of my websites. I have one at MIT and I have the at stephaniesenef.net. So I can put these slides up on both of those sites.
Okay, thank you. So when you have to go, I think it's about the end of your, or do you still have time, Stephanie?
I can stay on for a little longer.
Oh, okay. Then we are. So anybody else has any more questions or suggestions on how to proceed in this problem? Raul, go ahead. Can we hear you?
Yes, I have a question. The glyphosate affect the synthesis of sialic acid hominidate because glyphosate interact in the enzyme acetilneurominic acid.
Que no esté traduciendo, pero quiero hacer una pregunta y no la puedo hacer al mismo tiempo. Could you please talk to us about the importance of sunlight?
Sunlight. Oh, yes. Certainly. I think I worship sunlight. I think it's so important. I really encourage people to get out in the sun without sunscreen, without sunglasses. I mean, I have a personal story with my eyes because I just saw the eye doctor. They said everything's great. I don't have cataracts. I don't have any problems with my eyes. And my eyes have gotten better ever since I stopped wearing my glasses 20 years ago. I just stopped wearing them. And it turns out I can actually drive. My eyes are good enough to drive. They're not perfect, but they're good enough to drive without glasses. So I never wear glasses. I have none of the various macular degeneration, all those problems. And I've been exposing my eyes to the sun all my life. and I get out in the sun a lot. So this is really interesting because people are like wearing sunglasses. You know, they put the sunglasses on the 2-year-olds. I want to yank them off because I think the sunglasses on the 2-year-olds can cause autism because it's so important to get the sunlight into the pineal gland, which is right behind the eyes. I'm a very big worshiper of sunlight, both for the skin and for the eyes.
And I think this really highlights as well that connecting back in with nature is where the healing is. We have sunlight, we have water. Stephanie explained about exclusion zone water. And I do encourage everyone to have a look into exclusion zone water because that's a really interesting pathway to make extra energy when our mitochondria are struggling, but equally it's coming back to the food, the natural resources that we have available, and then we have our supplements and our other kind of nutraceutical interventions, and together they can form a really important intervention protocol.
I wonder if you can hear me now?
Yes?
Yes.
Oh, finally. Sorry. Yeah. About 5 years ago, Dr. Senef, you lectured a conference in one university in the United States regarding the importance of the sunlight, infrared energy, and the production of endothelial nitric oxide synthesis. It's pretty important because just having a sunbath decreases the hypertension or the
it can really drop your blood pressure. Yes, because it releases nitric oxide from the skin, and that relaxes the blood vessels and lowers the blood pressure. Yes, definitely, sunlight lowers blood pressure. And there's also the sulfate because sunlight, and Gerald Pollack showed this, that infrared light. So infrared light and UV light are both important. Infrared light actually grows the exclusion zone water by a factor of 4 in the experiments that he did. That he was really impressed to see that the infrared light would actually trigger the growth of that gelled water. The structure of the crystal would grow in response to infrared light. So that's quite amazing.
Does UV light does the same pattern? No.
Well, UV light may also, but infrared was the best at doing that, at growing the exclusion zone water size.
And faithfully decrease the blood clotting, isn't it?
Probably, yeah, I would think so. Yes. Well, the nitric oxide, of course, relaxes the muscles and allows the blood to flow, reduces the blood pressure.
And increase the sightseeing, right? The vision.
The vision, probably.
Yeah, it
I'm
will
not
increase
sure.
also. It will increase the flow of the blood in the eye.
Probably so. I mean, it fascinates me that we are told to buy sunglasses to protect ourselves from the sun. And sunglasses appear to make your eyes worse, in my opinion, because I'm one example. So maybe I'm not, representative of everybody, but I'm impressed that it defied the sunglass idea my whole life. And here I am at, quite an elderly age, so to speak, and still good eyes. I have all kinds of friends who are in much worse shape with their eyes than I am.
Maybe one day we can invite Dr. Jack Cruz to this forum because he is very knowledgeable on sunlight and all the other effects. But we have one more question, if I may, to Stephanie. It's from Pierre van Nucker from South Africa. Hi, Pierre. Good to see you here. And it discussed the heparin sulfide binding of the spike protein and what this could trigger in the clotting cascade. Stephanie, would you know about that? The heparin sulfide?
Well, I know the spike protein does bind to heparin sulfate, and that's sort of how it gains entry into the cell. But of course, that makes it sound like you don't want to have heparin sulfate, which I don't think is true, because you need to have the heparin sulfate to protect the cell to block the entry, because it creates the gelled water that is exclusion zone water. So it keeps things out. And so I think if the gel is not strong enough, then the spike protein can hook on. Otherwise, it gets blocked. I suspect that it's an issue of inadequate gel that allows it to reach in.
Maybe I may add the new versions of Omicron are very prone to bind to the glycocalyx, to heparin sulfate. But I don't think that they are able to really degrade it. They will punch a little here and there, prick some hole in there, but they're not a real problem for that. The degradation of the glycocalyx during COVID is actually owed to around 15 different pathways, which are the metalloproteinase, TGF-beta-1, reactive oxygen ROS, LPS, other toxic metabolites from the gut, and the macrophage activation and other factors. That is what, and the heparinase upregulation, that is what really causes then the degradation of the glycocalyx, and then the endothelium can be really affected, and then you have thrombi formation actually after that. That is really the consecutive order how SARS-CoV-2 spike protein gets into our endothelium and causes many of these coagulation cascades to form. Yeah, but the heparin sulfate is actually, I mean, you see heparin as a treatment to chelate spike and to work as an anticoagulant, so to speak. And we are using heparin analogs like fucoidans, which are heparin sulfate analogs also. And so you can give them into the organism during or after COVID in order to restore that the homeostasis in that sense. So they bind spike and they're like a decoy. Yeah.
Exactly. Yeah, I was going to use that word decoy, because if there's free heparin, it binds the spike can't bind the heparin sulfate that's on the glycocalyx.
Building heparin sulfate is a good thing. And to look, now we see so many people walking around with spike protein, and they have no symptoms. They are still healthy, so to speak. Non-measured, no biomarkers are off yet, but the persistence of spike is there. And when they start to degrade their glycocalyx and some other factors would occur, then all of a sudden you see some people tilt all of a sudden. Out of a healthy state, they go into long COVID or post-vac. Yeah.
Wow.
What you were telling today is the maintenance of the glycocalyx is essential. It is one of the things that can protect you from getting sick or chronically ill from exposure to SARS-CoV-2 and or spike protein. So I would emphasize that strong. Eat a lot of heparin sulfate containing foods. So we were even before COVID, Stephanie and myself, we were looking at all these chondroitin sulfide, hyaluronic acid, all the sulfate supplements in order to counteract this. And because it's, like I said, it's not only a detox thing, get this toxin out, you have to replenish the cascades that were brought down by this. And because it's and hand in hand with the GMO food and the excessive farming on the land is the decline of minerals, trace minerals, different proteins in the food. So you have not only an intoxication, you also have a deficiency at the same time, causing these problems in a concert. So rich food and take supplements they're not so expensive for some minerals and trace minerals and whatever vitamins are missing as a bridge maybe, and even butyrate, like you said, as a bridge. I would say it's not in the long term, but the SARS-CoV-2 spike is doing a massacre on the butyrate producing bacteria, and as is glyphosate. So for a while to take some sodium butyrate would be a good idea, just as a kickstart to get until everything else is up and running again.
Can I ask Pierre Van to ask his question? He's been trying to ask something.
Yeah, I think we discussed it right now. Thank you. Hello, Stephanie. Hello, Joachim.
Hi.
Yeah.
My question on the heparin sulfate was the heparin binds to the receptor binding domain of the spike protein, so it forms clotting. If this happens in your brain, it's a very bad place to happen. So I know that we need to have the heparin sulfate high, but it's also used in extracorporeal electrophoresis, Beate Jäger in Germany, to remove it outside of the body, to bind the spike protein.
To get the spike protein out, right?
Yes, it's the level that you need. It needs to be in the sweet spot, not too much, not too little, just right. So like the three bears. So that's the problem that I see, is that it can actually form clotting if it is too high in the body? That was my question, yeah.
I think there is a kind of paradoxical reaction, right, that people can have to heparin treatment, that it can cause the opposite of what it's supposed to protect from, and namely it can cause clotting when it goes wrong. And I don't know the process of that, but it's a tricky thing because the heparin, heparin is acting as a decoy to prevent things from binding to the heparin sulfate. That's sort of how it's working. So it's going to grab onto these things that are toxic and hopefully remove them. That's my understanding.
I also think that the data that we have on heparin sulfate analogs, especially for spike detoxification, is pretty promising, because by binding to the RBD and to the spike protein, let's say critical sites, you are kind of neutralizing the binding affinity of the spike protein, and they're very small. They're like I think less than 10 nanometers. So that is, you would need an electron microscope to see a spike protein. Not even they're visible. They're really small. So if you have a low molecular fucoidone or heparin sulfate, I think it's a pretty good idea to have them bind to the spike protein, and given the opportunity for the body to excrete it through the kidneys, liver, or any other means. I think it's a smart thing to detox or to, but that's another topic today. Yeah? But it's a smart thing to neutralize the spike and take away its binding capability to our host cell receptors in general. So I don't know. I think it's maybe also a question of dosage, of course. But I can check on that, Pierre. I will write you back. Let me check if there's any chance that heparin sulfate binding to the spike could trigger a conglomeration leading to thrombosis. I think it's not possible. I think it's many other pathways leading to that. But let me check and I'll get back to you.
Okay, thank you very much. And I can see someone from Jose Armando asking, please comment on apple cider vinegar. Any advice on?
Yeah, I like apple cider vinegar. I take vinegar every day. We always have a salad with salad dressing, organic salad dressing, homemade, and using apple cider vinegar. It's I have a hypothesis that it could be breaking down glyphosate enzymatically, because the Acetobacter, there are strains of Acetobacter that are able to break down glyphosate, but this has not been proven as far as I know. So it's only a hypothesis.
Okay, thank you very much. So can we come towards the end of the session, a wonderful session? Raul, would you like to?
Yeah, I would love to give back the word, the microphones to Dr. Stephanie Seneff, in case that she would like to address to us a final message regarding this session. What would be our homework, our take-home message, Dr. Seneff?
Well, I think, then is sunlight exposure, very important. Emphasize for your patients to get out in the sun without sunscreen, without sunglasses, and eating certified organic food, I think, is absolutely essential. And then avoiding processed foods. Don't eat things that come in boxes. Eat things that you can see, the cabbage and the broccoli and the shrimp, things you understand as animal and vegetable sources of nutrition, and cook at home. And spend the time, extra time that it takes to eat well. I think that's food is medicine, and I think the real path to good health is to eat well.
Yeah, as well as exercise.
Exercise too. That's true. Yes,
right.
Important to just take a walk every day. That's a really good thing to do. And of course, walking along the seashore is excellent, because if you've got some waves, you're going to get some depleted water to breathe into your lungs.
Yeah. Joachim Gerlach, final message, take-home message, or an idea, as well as Rachel and Dr. Susan Rush?
From my side, thank you everybody for coming in today, and thank you, Stephanie, for again highlighting these interesting pathways and giving us an inspiration on what to do with our own lives, families, and for the doctors with their patients. Thank you, Sarah, so much for coming in.
Thank you, Rachel.
Yeah, thank you again from myself, Stephanie. I mirror, and align with everything that you talk about with regards to putting simple, really simple interventions in place that people can implement as foundational tools to support their health. So thank you so much, Stephanie. This has been really helpful and very thought-provoking as well. I'll be coming back with lots of ideas, I'm sure.
Great.
Thank you, Dr. Rash.
Thank you. Thank you very much, Dr. Stephanie. Such a pleasure to have you with us, and it was enlightening. And we all will take home that, yes, our detox system has to be kept intact all the time. And there are so many methods, and every methods are important. Thank you. And we look forward to hear from you again and again. Thank you very much.
Thank you.
Dr. Nasser is somewhere with us here. Please, the microphones are open. Jose Nasser or Janine Galizia, if they are unable. Kerry. Final word to Dr. Seneth and all our audience, Kerry.
Well, this is always so interesting, and I get really excited when there's new stuff about autism. Sorry, I just laid down. But this is really, really interesting. And of course, you're always on the cutting edge, and everything you ever say has come true, unfortunately, with autism. But you've been warning for, over a decade, and the things are very important. And it's great to see two of my favorite people here, you and Joaquim. Thank you for everything.
Wonderful.
Thank you.
It's
Dr.
great
Yamashita.
to see you, Carrie.
Thank you.
Dr. Yamashita.
Thank you, Dr. Senef. It's such a great pleasure to have you here. It's an honor to have you again, and I hope you can teach us more things along this year, and see you again. Nice. Thank you very much.
Wonderful. Thanks.
And I see here Karen Johnson from New Zealand, who happens to be a very, let's say, a person who study quantum mechanics and quantum biology. Doctor, are you here with us?
Hi.
Yes, you're here, Dr. Johnson. It's nice to see you.
Good morning from New Zealand. Yeah, I'm still in my pajamas. It's early here in New Zealand. Yeah, look, I just popped in to say hi, and I've recently returned back from Spain giving a presentation on the helium Bose-Einstein condensate singularity model at an international summit on gravitation, astrophysics, and cosmology. We're really kind of building science at a new foundational level so that we can understand the principles behind how atomic clocks are operating in living systems. So these are the unstable atoms, and it's really connected to how consciousness operates. The memory formation, memory recall connected to the unstable atomic system using hydrogen as the key kind of mediator, and that connects into gene expression. I'd love the opportunity maybe in some future event to discuss the kind of implications of the new science. Really, really exciting information coming forward in terms of learning the underlying functionality of hydrogen in our biology is really shifting the perspective away from carbon and into a more dynamic, responsive, and it's utilizing time, right? So that's the paradigm change, is rather than seeing just structurally, we're seeing the relationship in the radius of the aromatic ring of dopamine, and the radius being 0.139 nanometers. It's very, very small. But when you convert that into meters, that's 1.39 e to the negative 10. and that correlates to the age of the universe, 1.39 e to the plus 10 years. So we have a functional molecule such as dopamine within our own physiology that acts really as a stargate to atomic clocks. So we're connected deeply to the field of energy around us at a second timing, giving us a positron-electron pairing system. And when you start to see this kind of implication, we are so far from being a physical being and more being this kind of cosmological phenomena connected to the entire universe through the functionality of these atomic clock systems that operate within our biology. And their timings are very strange because what they do is have a kind of dynamic dance with the future, and their half-lives really link our biology at the moment to future events in our lives. And they are kind of really navigating the complexity of our unconscious, linking our newly evolving and growing inner being of atomic energy to future events. And, you know, the strangeness of quantum biology is that it's not about the here and now, it's really about the future and our evolutionary path inward at a subatomic level. And so this new science that's really being built off new foundations, a foundation where there's a positron associated with every neutron in atomic theory, we've got a whole new model that shows us really the steps that our biology is going through to create our future selves within ourselves and how that emerges over time. So I know that's a little bit of a mouthful, but it's really showing that we have really in our own hands, within our own capability to understand our evolution within ourselves, and the physics within the aromatic ring based on atomic clocks is such a new and very strange biological system, but it's connected to a proton tunneling process. And it's a little bit like looking at black hole singularities. You can't really look inside the aromatic ring until you break it open with a hydroxy radical system. And when you do that, you kind of get a prophetic revelation of what's going on inside. And, you know, I discovered that when I had my own experience back in 2013, and it's those higher energetic processes within the body that lead to spontaneous remission from the likes of cancer. So the healing that happens within ourselves by breaking the ring open of this aromatic ring system associated with dopamine is that we have this capacity within ourselves for healing and regeneration that is natural. And it's growing high energetic states of unstable atoms as atomic clocks, and they provide us with this internal energetics to give healing and regeneration. So it's a very, very big paradigm shift that we're kind of all going through at the moment. And one of the reasons I think that the governments globally have unleashed COVID and the bioweapons on humanity is because we are discovering these very foundations to our own healing and regeneration. And once we have this internal power and the connection to the source of creation within ourselves, we're no longer, needing a nanny state, so to speak. We are empowered to be the creators that we are. So I'll leave it there, but I'd love to share more of the science and the detail behind it. Thank you,
Dr. Sheridan. Of course, you will be invited very soon. Let's schedule your next conference. So today it was a very special session for many reasons. The first one for your presence, the second one for our special guests, and finally for so many new interpreters joining to our team at DFW. Isabel Marquez from English to Portuguese, Mohamed Tayel Alusban from English to Arabic, Ishita Saxina from Portuguese to French, and also Shivani Dev from English to French. In my case, I try to do my best from English to Spanish. And additional thanks to Donna G. Rodriguez, nutritionist Maria Fernanda Uribe, Frida Rosas, Vivian from Mexico, Vivian Fleitas from Paraguay, and Patricia Aguiwa from Peru for their immense daily help to our organization. They represent a new generation of professionals committed since a very young age to their communities and the world's populations. A special thanks to IT specialist David Garcia from the College of Sciences and Humanities in Vallejo, who edited the DFW opening countdown and organizational video that of course, for technical reasons wasn't displayed today. Additionally to Norberta Rincon, who supported us or supporting us with the registration of the Vimeo DFW platform account. They are a source of pride to us. Thank you, Dr. Senev, Dr. Rash, Rachel, Joachim from Germany. Any final comment that you would like to address, Rachel?
Yeah, just to say, please make sure that you visit us on our social media platforms. We're on Facebook, Twitter, or now X, Instagram, and LinkedIn. And if you would like to become an active member and receive more news and benefits from the Doctors Federation for the World, you can go to our website, www.doctorsfederationfortheworld.org. On behalf of all of us at the DFW, the steering committee, staff, translators, everyone from all over the world, thank you for your presence and questions at this session, and we wish you a wonderful weekend. Goodbye.
Thank you, Rachel.
Thank you.
To everyone. Thank you indeed.
Bye.
Bye-bye.
Bye.
Have a nice weekend.
Bye.
Goodbye.